I've never in that situation. I can't do that. Okay. So, beginning today, we left off last time talking about lipids. So we went through our carbohydrates, which are essentially our sugars, and our big polysaccharides, which will be things like starch and cellulose that we'll talk about a little later on. Into our lipids, which are in general our non-polar hydrocarbons, lots of nitrogen, lots of carbon. We talked about triglycerides as our first group of lipids, so our fats and our oils. We left off with a short note about phospholipids, which is where we're going to start today. So hopefully you've seen our different biomolecules in previous classes. So I want you to do your best based on a little bit we did at the end of the class, as well as what you remember from your prerequisite courses, to draw a phospholipid or two. And I want you to think about how it orients with respect to water. So that's going to be super important as we start to talk about our cells. This is on the basis of our cell membranes. We've spoken about where we find water in the body. We know some of it is inside of cells, or intracellular fluid, and some of it is outside of cells, or extracellular fluid. So we want to make sure that we understand how these things are contained. And we're going to talk about how we regulate what goes in. I'm going to run someone else's office and grab a rock for activity later in class, but I'll be right back as you're trying to talk. Yeah. Oh, yeah. Okay, great. I agree some talking. So hopefully that means this was at least something that we can get a start on fairly easily. All right. So when we're thinking about our phospholipids, let's go ahead and begin essentially where we left off. So when we're talking about our phospholipids, one thing we know about them, and this is what makes them a lipid, right, is that they do have hydrocarbon chains, right? So the hydrocarbons are located in what we call the tails, the fatty acid tails. So the fatty acid tails are essentially the same as the fatty acids we saw in our triglycerides, right? So we can draw them pretty much the same, our little squiggles, right? So all this is carbon at every point, assuming that we have hydrogens coming off there. So those parts of our phospholipid are going to be non-polar because there's no real difference in electronegativity between hydrogen and carbon. You can also just remember that that's not polar. Okay? So this means they're going to be hydrophobic. They're going to repel water. When we're talking in general about biomolecules, there are certain atoms we can use as like shorthand to know if something's likely to be polar or non-polar. And really the main one that we see over and over again is oxygen. Oxygen is highly electronegative, so it is going to kind of hog electrons and cause charges to accumulate. Shorthand, if you don't totally remember talking much about electronegativity in the past. So fatty acids are just hydrogen and carbon, no oxygen, so we don't have little charges accumulating. But reminder, our water molecules, right, H2O, so we have an oxygen and we have two hydrogens. So water itself does have little charges accumulating. So oxygen is accumulating little charges because it's essentially hogging electrons kind of coming from those hydrogens. Okay, so you can think of as like a little kind of charged cloud here. Okay, that's not the official super chemistry explanation, but I'm a biologist and this is an A&P class, so that's sufficient for what we're thinking about here. Okay, so water in general is going to interact with things that are polar, so things that also have charges. And we could also think of these hydrogens as essentially having little positive charges kind of near them, right? Opposite charges attract. So anything that has like a little positive charge is going to want to be near the oxygen. Anything that has a little negative charge is going to be near this hydrogen side because they're like magnets for each other. Okay? Our lipids that are just hydrocarbons, so just carbon and hydrogen, don't have those little charges. This is why they repel water, essentially. It's because they don't interact with this environment well. So that's just backing up if you don't remember polarity. Okay. So our fatty acids, non-polar, hydrophobic, they don't interact well with water. Okay. So that's one region of our phospholipid. That's our non-polar tail here. But we also have our phosphate head. Okay. So this big round part is a phosphate group. So this part is polar, so it does have little charges accumulating around it. So it's going to orient itself near water. It's hydrophilic because those properties match well. So it's going to act like a little magnet essentially for water. So this means that if we're looking at the properties here, okay, so our polar head is hydrophilic. Okay, likes water, attracts water, is attracted to water. Our non-polar tail is hydrophobic. Okay. So it is going to try to escape water. So if we were to draw water around our phospholipid, we would expect the water to be accumulating kind of near the phospholipid head, that polar part, that hydrophilic part. And we're not going to expect water to be near the fatty acid tails. Okay. They're going to have ways that they escape water. If you just have one phospholipid, like one single molecule in a cup of water, kind of tough luck. But when we have multiple phospholipids, we've got some options. So questions about those properties of the phospholipid. So that is partially going to be forming the basis of how our cells work, about what can cross, what can't. How do we think about water? How do we think about ions? So that is an important thing to understand, is that chemical property of polarity and how it relates to charges and to water. We will talk about it more, but review that if you don't remember it. So one phospholipid in a cup of water, not much those fatty acid tails can do to escape the water. But we're pretty much never going to have just one phospholipid molecule. We're going to have a bunch of them. When we have a bunch of them, we can do something. Okay. So we know that we have our phospholipid head, water near it, so we're gonna have some water here. Just drawing what I was drawing on the previous slide. Okay. Our fatty acid tails, I said, don't want any water near them. So if we have multiple phospholipids all together, the way they can do that, a way they can protect themselves, is essentially by forming a shell of those polar heads, of those phosphate groups, facing whatever water exists, and group all those fatty acid tails together. So that's what we see happening here in this structure called a micelle. So this one, we just group them all with those tails facing in, right? There's no water in the center here. And then all of our polar heads, all of those phosphate groups are on the outside. Okay. And this blue part is our water. Okay. So those fatty acid tails here are protected from the water. However, when we're talking about a cell, we have extracellular fluid, right? So the stuff outside the cell, outside, right? Extracellular fluid, ECF, would be out here. But a micelle doesn't have any water inside it. A cell does, because we also have intracellular fluid. So on this image, imagine that we've cut through, essentially, I'll use different colors so I don't reuse blue too much. Okay. So this orange border, my cell, outside, let's say we have extracellular fluid. That's what's outside of cells. Inside our cell, we have intracellular fluid. Okay. And this image on the left, we've essentially taken just like a slice here, right? So this is where we would find in a cell our phospholipid bilayer. So it has to contend with, it has to adjust for the fact that there's water outside in the extracellular fluid, and there's water inside in the intracellular fluid. Okay, so our ECF, we can draw at the top. ICF we could draw at the bottom, but that top bottom orientation doesn't matter much. This is just kind of standardizing things to make it a little easier to think about. Okay. But what does matter is that those fatty acid tails want to protect themselves from all of the water, whether it's outside or inside this cell. Okay. So this is why we end up with a bilayer, bi meaning two, layer meaning layer. Okay. So what we've done is we've oriented some of those polar heads, so those phosphate groups, out facing the water in the extracellular fluid. We can draw a little X's for our water molecules here. But if we only had one layer, that would leave our fatty acid tails touching the intracellular fluid, and they absolutely do not want to do that. They're hydrophobic, they're afraid of water. So what happens instead is that we end up with a second layer that's in the reverse. Okay. So here we have some more polar groups, some phosphate heads. Okay. And in the center, our fatty acid tails of one phospholipid, okay, are just facing end to end the fatty acid tails of the opposing phospholipid in the opposite layer. Okay. So here we have essentially two phospholipids upside down, right? So they're putting their feet to each other. Okay. So this means that we always have a polar hydrophilic phosphate head touching the water, and those fatty acid tails are always protected in the center here. As we start to talk about the properties of cells and membranes, we'll want to note then that this creates a big non-polar hydrophobic region in the center of our membrane. Right, so I'm going to write non-polar and hydrophobic. Hydrophobic. But I can't abbreviate that. Okay. Cool. And our hydrophilic part is thinner because it's just those polar phosphate heads, but we have a little bit up here and a little bit down there in the bilayer. Questions about the structure of the phospholipid or about this kind of like bilayer formation? So after today, if you're confused but don't want to tell me right now, think about it, let it percolate. Think about whether you have any questions, because understanding this is going to be important to understanding what can and can't cross our cell membranes. So we're thinking about those chemical properties. So the main place we'll see phospholipids are in the membranes of our cells, but we will also see phospholipids elsewhere when we talk about the respiratory system and the lungs. Another phospholipid that we see is something called surfactant. Surfactant is a phospholipid you have specifically within the tissue of the lungs that prevent them from collapsing on themselves, essentially. So there are multiple types of phospholipid. The main one we think about is this phospholipid bilayer. I don't think that's true. Okay, so I will send you updates about SI. That's a great reminder for me. Unless, did they send out an email about SI already separately? Okay, great, because I gave them access to our workspace. Awesome. All right, so in that case, what I will remind you is that for SI, we do want you to be actively studying for this course throughout the semester throughout the unit. So we'll give you 1 point of extra credit every week you attend. So I've asked the SI leaders to share with me that list every week. So if you can't make those hours, I believe they put up the worksheets and have a separate system for tallying that. But if there are any problems, the point is for you to engage extra with the material. So if you talk to me, we can set up something as an alternative arrangement, maybe talking briefly during office hours, something like that. So let me know if there's a problem. Okay. So we're not done with our lipids yet, although the phospholipids, I would say, are really the most important thing to know and understand. And the triglycerides are the one you're most often going to see as the stereotypical example of lipid. However, in the human body, we have one. So our third class of lipid is going to be an eicosanoid. So we can recognize this based on all the hydrocarbons we're seeing. So carbon, hydrogen, carbon, hydrogen, carbon, hydrogen everywhere. That should tell us that this is mostly going to be non-polar hydrophobic. So this is what makes something a lipid, is that property. These are essentially modified fatty acids. So they're like fatty acids kind of folded over on themselves with a couple extra steps. So think about it just kind of folded in half like that. I'm not going to ask you to memorize those examples, but what you do want to be aware of about this group of molecules is that this essentially acts as a signaling molecule in your body. So we really won't speak about them for the rest of the course, but as you move forward in your careers, it will probably come back as you think about intercellular communication. Okay. You'll think a fair bit more about steroids. Okay. This is the place to remember. In our previous three, we saw something that looked vaguely like a fatty acid. You don't see that so much if you're looking at the chemical structure of a steroid, but it's still a lipid, and you can still recognize it's a lipid because it's just carbon and hydrogen everywhere. Okay. So at first glance, if you're not being careful, those rings might remind you of our sugars. They might remind you of our carbohydrates, which was why I pointed out when we talked about the carbohydrates that they look like they're kind of holding hands with an oxygen in the middle. Right? You can see the steroid ring structure is really just made up of carbon and hydrogen, and those rings are pressed against each other. It's slightly different, and it's based on essentially these 4 rings. So it's always kind of going to be 4. You may have something else kind of hanging off of it, but you should be able to find this basic 4-ring structure within a steroid. So steroids are important when we think about steroid medications, but they're also produced naturally by your body. So many of our hormones are based on steroids. So steroid hormones and therefore lipid hormones be things like estrogen and testosterone, sex hormones, also aldosterone, which we'll talk about in A&P too. All of them are derived from cholesterol. You might have thought about cholesterol when you're thinking about your diet. There's good cholesterol and bad cholesterol. And when we get to our cell membrane, we'll notice that cholesterol is going to be one of the components of the cell membrane in humans. Okay. So questions about lipids at this point, because we're going to move forward to our proteins. Our proteins are going to do a lot of things in the body. And they're going to form really long chains that essentially fold around themselves in order to have all these different functions. So proteins are partially so varied in their activity because they have complicated three-dimensional structures that can vary a lot more than some of these other molecules. Okay. I'm not going to rip off 20 pieces of tape, but what I am ripping off right now are going to be a couple amino acids, which are going to be the building blocks of my proteins. Okay. So I have two colors. I've got black and silver up here, but we actually have 20 different kinds of amino acids, okay, that are going to form the building blocks of our complicated proteins. So things to know about our amino acids before we even get into how they bond together. Okay. When we look at an amino acid, we're going to see a stereotypical structure. So in the center, we have a carbon. Okay, so it's an organic molecule. Below the carbon, you're going to see just one hydrogen. And then on either end, we're going to have a carboxyl group and amino group. So that's true for every amino acid. So here we have highlighted a carboxyl group, and here we have highlighted our amino group. When we think about the different amino acids, what we're going to notice is that here we have an R. So R isn't a specific atom. R is just a variable there. So you can think of it as like, I don't know, sometimes I think R for replaceable, right? This is our replaceable group. Our 20 amino acids differ because they have a different group there in that R position. Okay. So different amino acids, our 20 different amino acids are going to have different chemical properties because we can substitute whatever we want there. We can substitute one of 20 things there. So that's the basic structure of an amino acid. So if you're trying to identify an amino acid from its chemical structure, those are the things you're going to look for. You're going to look for that nitrogen, which we haven't seen yet, right? We didn't have nitrogen in our carbohydrates. We didn't have nitrogen in our lipids, right? And we can see that carboxyl group. And as we start to think about how our amino acids bond to each other, our amino acids, they get next to each other, so link them up, they're going to form a peptide bond, right? So amino acid 1, be this silver piece, amino acid 2, this black piece of duct tape. When I link them together, I've just formed a peptide bond, okay? So that's the first part of building a protein. And it just continues like that. You're going to continue building. I want to make sure I'm alternating my colors onto the end, forming more and more peptide bonds as you start to form like a polypeptide chain. Now, this means that if you are looking at a protein or a polypeptide chain, you're going to see along its backbone in the center a pattern that goes nitrogen, carbon, carbon, and now you're moving into where the next amino acid was. So nitrogen again, carbon, carbon. So if you're looking for that nitrogen repeating over and over again, that will also help make sure that you're not looking at a lipid, right? So when you're looking at that chain, be careful that you're looking at what's inside of the chain. Now we have different words for amino acids linked together in certain numbers, essentially. So when we have just a small little chain of amino acids linked together, and by small, we mean about 2 to 50, we're going to call it a peptide. When we get really big, so more than 50, and sometimes a lot more than 50, this is when we're going to start to form proteins. Questions about this first step of formation of the proteins? Okay, cool. So we got this covalent bond between the carboxyl group and the amino group for making chains of amino acids into peptides, and then once we get big enough, into proteins. Okay. Proteins get complicated because they have several levels of structure. So the primary level of the structure is just that chain. So primary structure is just which amino acids you have in order. So they're going to be joined with peptide bonds, just in a line. Okay? That's the primary structure of our protein. But proteins can do so many things because they have three-dimensional shapes. So then we start to have three-dimensional structure eventually. So before we end up with a big complicated shape, we start to form one of two shapes in different parts of our chain. So we can form alpha helixes or helices. So helix is like when we get to DNA, right? That structure of DNA, so like a spiral staircase structure. Alpha helix, alpha for like option A. The other option we can form are beta pleated sheets. So think about like a pleated skirt. That's just like a zigzag of this sheet. Okay. So that would be the secondary structure. Okay. And this is held together by hydrogen bonding. So you can see it's just because there's an attraction between oxygen and hydrogen, basically because of those little particles building up on oxygen. So that's how we start to crumple up our protein. And then the tertiary structure, where it starts to get more interesting. So the tertiary structure, right, we made an alpha helix, or kind of swirled this around. Now we're going to start to have interactions between those R groups. So we're going to start to like essentially crumple this up into a ball. This is a very sophisticated prop I made for you guys. Okay. So this is our tertiary structure. Our crumpled up ball or crumpled up piece of paper. So you can see within it, we have some of those secondary structures from the previous slide. So here you can see a nice curly alpha helix. Here you see that back and forth beta pleated sheet. But you can see it's more complicated here. Okay? So we've started to crumple up. We started to have R groups interact with each other. And we're not being more specific about that because there are 20 amino acids. So we have 20 options of what types of chemical properties they might have. And it's really going to vary. But you can imagine, say, let's look here. Okay, so say we had an R group here and an R group here that had the polar properties, like one is positive and one is negative. So that's why they joined up real close to each other, right? That's why they started to crumple in that form. And we have different interactions going on between other parts of this chain elsewhere, depending on other R groups. Finally, our quaternary structure. So notice we've just gone 1, 2, 3, 4 in slightly fancier words, right? Primary, secondary, tertiary, quaternary, like quad means 4. So our quaternary structure is when we have several crumpled up balls. sticking to each other, right? So we have two subunits that get stuck to each other, and that's our quaternary structure of the protein. So what we're looking at here is hemoglobin. So hemoglobin carries oxygen in your blood. So here we have one subunit, two subunit, and then in back, that's why they're paler, 3 subunit, 4 subunit. Okay. And so these are going to be attracted to each other depending on what chemical properties the R groups facing the outside of this bundle are, right? So what I did when I stuck these together, right, there's a sticky bit on this crumpled ball, there's a sticky bit on this crumpled ball. So I put those together, right? It wouldn't really matter what's on the inside in order to create this. So this is still R group interactions, but we really think of them as subunit interactions because we might think about these different pieces separately. So this would be a subunit. So questions about the overall structure of the proteins and how they form these three-dimensional shapes. Hopefully we'll get to enzymes by the end of today. If not, we'll get to them on Friday. But before we get there, when you think about how these proteins are forming their structure, that kind of explains how the enzymes work as well. So enzymes are biological catalysts that are going to speed up reactions. But in order to do that, they attract things, right? So if you think about this outside of this protein as having chemical properties that can act as a magnet for something, that's essentially how our enzymes are going to attract things as they start to set up, preparing to catalyze chemical reactions. So we'll talk more about enzymes when we get to the enzyme slides, but that's just kind of linking it up here. So do remember that enzymes are proteins. But we're going to round out our biomolecules, our macromolecules, by thinking about our fourth class, our nucleic acids. Okay. So nucleic acid, you're going to abbreviate NA. We're going to have essentially two types of nucleic acids we think about. So we're going to have DNA, and we're going to have RNA. So these are going to contain the genetic code in the form of DNA. The RNA is going to help us read that code and build proteins out of it. So DNA in its sequence codes for specific amino acids that then build a protein that crumbles up and does something. When we're looking at this structure, we are going to see that a nucleic acid has a carbohydrate inside it. Right? So it has a ring here, a sugar ring. So here's that carbohydrate bit. It has a phosphate group off to one side. And then it has a variable nitrogenous base on the other side. So you can think about it like standing and holding out an arm and holding out an arm. So phosphate group over here, our 5 carbohydrate ring. You can count them 1, 2, 3. I should start here. 1, 2, 3, 4, 5. Okay. And then those bases. The difference between DNA and RNA in terms of their chemistry is what carbohydrate is inside. So that carbohydrate is just labeled carbohydrate in the general form. But for DNA, we're going to have deoxyribose as our sugar, our carbohydrate. And for RNA, we're going to have ribose as our carbohydrate, our sugar. Okay. Deoxyribose and ribose look very similar to each other. But if you're looking at them, You can kind of tell from the name what's going on, which one you're looking at, because I'm going to start on the right with the ribose. So at the bottom of the ribose, you can see we have 2 hydroxyl groups, 2 alcohol groups, whatever you want to call them, the OHs. OH on the bottom, OH on the bottom. Okay. On the deoxyribose, deoxy, think we're taking away an oxygen. Okay. So we have 1 OH group here, but at that spot where ribose has another OH group, we just have a hydrogen. That's really all the difference is between ribose and deoxyribose. That leaves us with our bases. Okay. So our bases are what we use to create the code for the amino acids. So our bases are what can vary because we have different options. We can put them in different orders to code for different things. Okay. So we have our pyrimidines. Okay. So we have cytosine, thymine, and uracil. Okay. So C, T, and U. We have our purines, which are adenine and guanine, A and G. Okay. So the pyrimidines, if we were looking at them, you just have 1 little ring in that base. The purines, you have 2 rings. Okay. We're not going to really think again about those words, pyrimidines and purines, except to note that DNA has a consistent thickness as it goes through. So this structure is part of why you have specific bases pairing to each other. Okay. This is not a genetics class, so we're not going to go through that much, but I'm just going to note DNA is 2-stranded, right? So we have bases holding hands with each other. But RNA is often just 1-stranded. Another thing that we might group in with our nucleic acids are our energy transferring nucleotides. So each of those individual little pieces that we saw, so when we say A, T, G, whatever, A would be a nucleotide, T would be a nucleotide, or standing for 1 monomer, 1 molecule. Okay. The 1 building block of a molecule. We do have nucleotides that aren't involved in forming DNA or RNA, but do have important functions in the body. The most important of those is something called adenosine triphosphate, or ATP. And who wants to remind me why we care about ATP? Exactly, energy. So ATP is going to be our form of cellular energy. So our next bit of this unit that we're going to move into is we're going to be thinking about metabolism. So we're going to be thinking all about where do we get this ATP from, essentially. Okay. So ATP, the structure, is an adenosine. So I'm going to look back. So our adenine was here, right? So that's going to be our building block for our adenosine triphosphate. So you can see that here, that nitrogenous base, that ribose sugar. But what we have holding onto it are 3 phosphate groups. So 1, 2, 3. Okay. So that's where the tri or the T comes from. Adenosine triphosphate. And we can think of it as holding energy within each of those covalent bonds between the phosphate groups. So here and here. If we break those bonds, we're going to release energy. So think of it as making like a big pop sound when you break one off. And to put them together takes a little work. You have to put some energy into that bond. Okay? So when we think about ATP, as we move forward, we're going to be thinking about how when we break off a phosphate group and just end up with 2 phosphates. I'm just going to go P, maybe attach there. Right, this is going to be a lower energy form. If we hold some energy in that 3rd phosphate group in that bond, we get ATP. So that's the difference between ATP and ADP. Okay. So ADP, the D is for di or 2. Okay. I'm pointing this out now. When we'll really want to think about this is when we start to think about muscles and how they use energy. We'll be looking at a cycle where we see the conversion of ATP to ADP, and then we have to go back basically and get more. Okay. So these are forms of nucleotides. When we're talking about metabolism, which we'll come to quite soon, we are going to think about 2 other nucleotides that also kind of hold energy for us. So NAD and FAD. We're going to think about in metabolism as being our electron transporters when we start to talk about the steps of cellular respiration. So they also go in this group. They work not quite the same as ATP, but they have a similar kind of role in moving energy around. We are not going to think about this next little piece in terms of its structure, but I did want to show you a picture just once. Cyclic nucleotides will be important messengers when we start to talk about processes that happen within a cell. And I'm going to just highlight this camp, cyclic AMP, CAMP. Okay, so this would also go within this group. And this is going to be important when we get a signal from outside the cell to tell a cell to start doing something internally. Cyclic AMP is often going to be involved. We'll talk about identifying certain things based on the presence of cyclic AMP or not. You don't need to memorize this right now, but I want to show it to you because we'll talk about it later. Questions about these basic classes of biomolecules, macromolecules overall. We want to be able to identify them. We want to think about some of their chemical properties as we start to think about what can cross a cell, what can't. So we're going to be thinking about polarity, so little accumulated charges. And spoiler alert, that's going to make it difficult for things to cross cell membranes. Okay. And we'll also think when we think about proteins, about size. Stuff that's too big, it's just not going to fit through our phospholipid barrier. You're going to have to squeeze between molecules. But we'll talk about that when we get to the lipids. We are going to briefly begin our enzymes. And pick up with this more on Friday. In a couple minutes. For our enzymes, first thing to know about an enzyme is a protein. So an enzyme is a group of proteins that have a specific type of action. So it's going to have that big folded up shape that we talked about with proteins generally. Their specific function is going to be to act as a catalyst in the body. So they're going to speed up chemical reactions. Lots of chemical reactions do happen spontaneously, but we do not want to sit around waiting for our cells to just spontaneously have molecules bump into each other and form and break bonds. That would not work very well. And we also wouldn't be able to control and kind of regulate and make sure that everything's going along at a kind of consistent speed. So that's why we're going to need enzymes in our cellular processes. The way they do this is they decrease our activation energy for specific reactions. So you're going to use specific enzymes to break apart peptide bonds. You're going to use specific different enzymes to break apart carbohydrate bonds. Right? Sometimes we can reuse an enzyme for multiple types of reactions. but it's not like one enzyme can do absolutely everything, okay? So they're gonna be specialized. Okay? So what happens when we're looking at an enzyme? Our enzyme, my 3D shape for my enzyme. Okay, so that's our big protein that's going to catalyze a reaction. It's going to bind to a substrate. Okay. It's binding into what we call the active site. Okay, so enzyme, substrate. Okay. When they're together for a while, they're bound. Right? Then the enzyme is going to speed up some reaction. So we're going to end up with our enzyme back again. And our substrate or our reactant is now different, right? So now I'm going to draw a triangle instead of a circle. So basically, if we wanted to turn that circle into a triangle, we could wait around, waiting for it to happen. We don't want to wait. So we're going to take an enzyme and make that easier by decreasing the activation energy for this reaction. So if we want to look at this in terms of more scientific kind of graphs, okay, we're going to think about our energy essentially here on the y-axis and our reaction coordinate. So basically like time over the reaction kind of on the x-axis. So since I'm being a little hand wavy, I'm not going to label them right now. But that's what we're talking about. Okay. What happens across the course of the chemical reaction? We have our reactants that start out with some amount of energy. Our reactants, or when we're thinking enzymes, substrates is the word we'll use. I'm going to use the chemistry words right now. Okay. We want to end up with our products. I'm going to draw them with less energy here. So because our product has less energy in it than the reactant, this could potentially happen spontaneously. You don't have to add in any energy, but you'd have to wait until that reactant starts essentially like kind of vibrating and bumping into stuff enough for it to become unstable and then take us down to the product. Okay. So that hump here, that lump, that rise, that's our activation energy. Activation energy. What our enzyme does, it lowers that barrier. So essentially you have to accumulate less energy for this to happen. I'm going to draw a blue line about what might happen with an enzyme. All right, so reactant, you need to get a little energy, and then we immediately just can go down to the product much easier. So if you have a lower activation energy, so if that little pump is smaller, things are going to speed up. They're going to take less time because you're not waiting around for that perfect condition to happen. So here it is with everything labeled out. So here we have a drawing, right? We're starting with our reactants with a certain amount of energy. Without an enzyme, they have a big activation energy barrier to get down to the products. With an enzyme, you only need a little bit of activation energy before you can move forward. So that is where we will leave off for today. And next time we'll be talking about substrate specificity. Note for you guys as you're reading the textbook. So I believe we're chapter 1 now. We got into chapter 2. Chapter 2 has a big section on organelles stuff. Right here. We're not going to really be covering organelles in our unit. You're good in general, or if I mention an organelle, have an idea of what that is. So go ahead and review them if you don't have a general idea. But you're not gonna be tested on them in this. That was shot. Actually, that's on. I think person that I don't know.